morning, good afternoon, and good evening to everyone who joins us from around the world.
It is wonderful to be here.
This is Andrew and Renee. Welcome to the Stacks series.
It is always a pleasure to unpack the research with you.
Today we are exploring microbiome engineering, longevity, and the future of personalized health.
And honestly, this is one of those topics that it really fundamentally changes how we see ourselves.
It really does.
It challenges the very definition of what it means to be human.
I was just thinking that. I was reading through the stack of research we have for today, and we've got some heavy hitters like genome medicine, nature aging, all that incredible data from the Weizmann Institute. And I kept circling back to this one phrase that just stuck with me.
Oh, yeah. What was that?
The invisible organ.
It's a powerful image, isn't it?
It is, but it's also a little unsettling, you know? We grow up thinking of our bodies as this singular unit. I'm me, I have a heart, lungs, a brain. But all the literature suggests we are actually something called a holobiont.
A holobiont. I know. It sounds like something out of a sci-fi novel.
It really does, or maybe a Marvel villain.
Exactly. But it's the biological reality.
Okay, so let's break that down immediately. I think that sets the stage for everything else we're going to get into today. What is a holobiont?
Well, the term holobiont basically refers to a host. So that's you, plus the billions, actually trillions, of microorganisms living in non-you.
Trillions.
Trillions. We're not a solitary individual. We are a walking, talking ecosystem. We're a coral reef in human shape.
A coral reef in human shape. I like that.
And for the longest time, medicine just treated this ecosystem as, background noise, something that was just there.
Right. I remember growing up, the only time anyone ever talked about bacteria was when you needed to kill it.
With antibiotics, yeah.
Or maybe if you were really on the cutting edge of health, you were in the eat some yogurt and hope for the best era.
Exactly. And that was the observational phase. We looked at the bugs, we gave them names, maybe we tried to, feed them with some probiotics we bought at the store.
Right.
But we are crossing a massive threshold right now. We're moving out of that era of just looking and into the era of engineering.
Engineering. That word implies design. It implies control.
It does. We are not just watching the microbiome anymore. We are learning how to program it.
Program it.
Yes. We're talking about turning bacteria into living, microscopic drug factories. We're talking about using them to extend the human lifespan.
And that's where the stakes get incredibly high. This isn't just about, curing a stomachache anymore.
Not at all.
The research here is pointing towards treating diseases that have baffled neurologists for decades, things like Parkinson's. And then there's this phrase that kept popping up, designing Methuselah.
Designing Methuselah. It's a loaded term. It essentially asks the big question, if we can design a microbiome that allows you to live to be 150, should we?
A huge ethical question. So let's try to unpack this. We have a massive amount of ground to cover. We need to understand how our gut ages because apparently it has its own clock.
It absolutely does.
And then how we can potentially hack that process using synthetic biology and what this future of digital twin technology actually looks like.
Let's start with the clock.
The clock inside.
The clock inside. So when you think of aging, what's the first thing that comes to mind?
Oh, the usual stuff. Gray hair, wrinkles, my knees make that popping sound when I stand up too fast.
Right.
Maybe forgetting where I put my keys for the third time in an hour.
Exactly. Those are all the external or maybe the cognitive signs. But there is a distinct, measurable aging process happening deep inside your gut, and the literature has a name for it, biome-aging.
This concept from the genome medicine paper was just fascinating to me. The idea that your gut ecosystem has a life cycle that's sort of independent of your own, but also deeply connected to your health. It's not static.
Not at all. It's not static. It follows a very specific trajectory, almost like a story with a beginning, middle, and an end.
So let's trace that story. Where does it start?
It starts at the very beginning. Think about infancy.
Okay, so a brand new baby. Day one.
Day one, or at least in the first few months. A baby's gut is, for the most part, a monoculture. It is dominated by one specific type of bacteria.
It's just one?
Mostly, yeah. It's called Bifidobacterium. Now, why do you think that would be? What's the logic there?
Well, babies really only have one thing on the menu.
Right.
Milk.
Exactly. The input is singular, milk. So the ecosystem is highly specialized to do one job really, really well. Digest milk, sugars, the simple, efficient economy.
Okay, so that's the start. Simple, specialized, then what happens?
Then you hit adulthood. You start eating solid foods. You crawl on the floor. You put weird things in your mouth.
Hopefully you stop eating off the floor at some point, but yeah, the exposure happens.
The exposure happens.
Okay.
And because of all that new input, diversity absolutely explodes. You enter this complex balancing act between two major groups of bacteria, or phyla, the firmicutes and the bacteroidetes.
And that's the goal, that balance.
For a healthy adult, yes. This is the prime of your microbiome. High diversity, which means high resilience. It can handle a bad meal or a course of antibiotics and bounce back.
So that's the peak. That's the healthy garden in full bloom. But then the slide begins.
Then comes old age. And the research shows a very distinct, almost universal shift. It's not just random chaos or decay.
What does the shift look like?
You see a significant loss of those beneficial microbes, the ones that kept everything stable. And at the same time, you see a corresponding rise in what we call pathobionts.
I want to pause on that word. Pathobionts. It sounds distinct from a pathogen, but it also sounds not good.
It's a critical distinction, and it's key to understanding this whole process. A pathogen is a foreign invader. Think salmonella or the flu virus. It doesn't belong there. It crashes the party specifically to break the furniture.
Got it. An outside attacker.
A pathobiont, on the other hand, is a resident. It lives in your gut normally, usually in very small, quiet numbers.
So it's like a neighbor who's usually quiet and keeps to himself.
Perfect analogy. When your ecosystem, your neighborhood, is healthy and diverse, the good bacteria keep the pathobionts in check. They crowd them out. They don't give them space to grow.
But when the neighborhood starts to fall apart...
Exactly. When diversity drops in old age, these pathobionts seize the opportunity. They overgrow.
It's like weeds taking over a garden when the gardener retires. The weed seeds were always there in the soil, but now they have the run of the place.
That is the perfect analogy. And this shift isn't just a quiet change. It actively drives a process called inflammaging.
I see this term everywhere in longevity circles now. Inflammaging, it's a mashup of inflammation and aging, right?
That is exactly what it is. But we need to explain the mechanism here, because it explains why we get so many different diseases as we get older.
Okay, let's get into it.
As the microbiome ages and this diversity drops, the gut lining, the physical barrier between everything you digest in your blood, starts to get permeable.
This is the leaky gut idea, right? I feel like that term gets thrown around a lot on wellness blogs, sometimes in a very unscientific way.
It does, and that's unfortunate, because in this context, it's a specific, measurable biological breakdown. Think of your gut lining like a castle wall. It's made of cells tightly packed together. Its job is to let the good stuff in, nutrients, but keep the bad stuff out.
And who's the enemy in this scenario? What's trying to get through the wall?
The main villains are toxins, specifically things called lipopolysaccharides, or LPS.
LPS.
LPS are basically just fragments of bacterial cell walls. They're endotoxins. When your gut wall is strong and healthy, they stay inside the gut and, well, they eventually leave the body.
Right.
But when you have a leaky gut, when those tight junctions between the cells loosen up, these LPS fragments slip through the cracks and into your bloodstream.
And once they're in the blood, they can go anywhere in the body.
Anywhere. And your immune system absolutely hates them. It identifies them as a major threat, a sign of invasion.
So it launches an attack.
Well, yes, but here's the key. It's not a massive invasion like a cut or a big infection. It's a slow, constant trickle of these toxins.
Yeah.
So your immune system doesn't launch a full-scale war. Instead, it just keeps the alarm bells ringing at a low volume, constantly, 20/47.
So you have this chronic, low-grade inflammation just simmering in the background all the time.
Exactly. And that chronic, simmering inflammation is the engine behind Alzheimer's, heart disease, metabolic syndrome, type 2 diabetes, basically all the major chronic diseases we associate with getting old.
Wow. Okay, so let me just try to summarize this to make sure I have it straight. The gut ecosystem has its own aging process.
Yep.
This causes the good, diverse bugs to die off and the bad weeds, the pathobionts, to grow out of control.
Correct.
This overgrowth weakens the gut wall, making it leaky. Toxins get into the blood. The immune system gets irritated and just stays irritated. And that constant irritation slowly damages our organs over decades.
You have nailed it. That is the standard trajectory of biome-aging.
That sounds incredibly depressing. It sounds completely inevitable.
It sounds inevitable for most people. But, and here's where the data gets surprising and honestly quite hopeful, there are anomalies.
The centenarians.
Centenarians, people who live past 100. And even rarer, the semi-supercentenarians who live past 105.
Right.
Researchers have been sequencing their microbiomes for years, fully expecting to see this classic biome-aging decline. They expected to see the weeds and the leaky gut and the inflammation.
But they don't find it, is that you're saying?
They do not see the same pattern. These individuals are, in effect, opting out of biome-aging. Even at 105 years old, they maintain incredibly high microbial diversity. They have what scientists are calling a youthful signature.
Okay, so they're the biological lottery winners. But have we identified who is responsible? If I were to look into the gut of a 105-year-old, who are the heroes, the specific bacteria keeping things running smoothly?
We can actually name names now.
Let's hear them. Who are the VIPs in the centenarian gut?
Okay, so first up, there's a family of bacteria called Christensenlacea.
Christensenlacea. Okay, that is a mouthful.
It is a mouthful. But what's fascinating about this one is that it's highly heritable.
Heritable, so you mean it's passed down. My genes determine if I have it.
More so than for many other bacteria, yes.
Hmm.
Your human genetics play a huge role in whether your body is a welcoming host for Christensenlacea.
Mhm.
And having this bacterial family is strongly, statistically linked to leanness and to extreme longevity.
So part of it really is just winning the genetic lottery.
A part of it seems to be.
Okay, so that's one. Who else is on the team?
Akkermansia muciniphila. Now, this one's getting a lot of press. It's often called the guardian of the gut lining.
I have heard of Akkermansia. Isn't this the one that
Yeah,
it eats mucus?
it is. It lives in the mucus layer of your gut, and that is its primary food source.
Which, intuitively, that seems bad. If I want a strong barrier, why would I want a bacteria literally eating my protective mucus layer? It seems like a paradox.
It does seem like a paradox, doesn't it? But think of it this way. Imagine your mucus layer is like a lawn. If you never cut the grass, it gets all thin and patchy, thatch builds up, it gets unhealthy, it stops growing effectively.
You have to maintain it.
You have to maintain it. Akkermansia acts like a lawnmower. By constantly grazing on the old outer layer of mucus, it stimulates the goblet cells. Those are the cells in your gut lining that produce mucus to make more fresh, thick mucus.
Ah, so it's forcing a constant renewal.
Exactly. It's like strength training for the gut wall. By stressing it just a little tiny bit, it makes the whole system stronger and more robust.
So it's not letting the wall get stagnant and thin, it keeps the defense active.
It keeps the defense active and thick, which is critical for keeping those nasty LPS toxins in the gut where they belong.
Okay, so we have the heritable one, Christensenlacea, and the guardian Akkermansia. Anyone else?
Yes, one more critical group, bacteroids. Specifically, a couple of species like B. thiotomicron and B. uniformis. But the key here isn't just that these bugs are present. It's not just a roll call of who's there.
It's what they're doing.
It is what they are doing, function over form. Centenarians have a unique ability to produce what are called secondary bile acids.
I know the liver makes bile to help us digest fats. That's primary bile, right?
Correct. The liver makes primary bile acids. They flow into your gut to help you digest a cheeseburger. But then something amazing happens.
What's that?
Your bacteria get a hold of those primary acids and they chemically modify them. They perform alchemy on them. They turn them into secondary bile acids.
It's like a little chemistry lab down there.
It is. And in centenarians, their bacteria produce a very specific, very potent secondary bile acid called isoallolithicolic acid.
Isoallolithicolic acid. Okay, that sounds serious. What does it do?
It acts as a sniper.
A sniper.
It is a potent antimicrobial agent. But it's not a broad-spectrum bomb like an antibiotic.
Yeah.
It specifically kills off bad bacteria. Specifically, it targets gram-positive pathobionts like Clostridium difficile.
Oh, C. diff. That's a nasty one. It causes huge problems in hospitals and for the elderly.
Huge problems. Terrible diarrhea, colitis. But this secondary bile acid produced by the centenarian's own gut bugs kills it on contact.
Wait, so their own gut bacteria are manufacturing their own targeted antibiotics to keep the weeds out of the garden?
Yes. It is a built-in self-cleaning mechanism. While other elderly people are losing their defenses and becoming vulnerable, the centenarian gut is producing its own personalized weed killer.
That brings us right back to that garden analogy. It really holds up.
It holds up perfectly. If you look at the microbiome of a frail elderly person, someone with low energy, poor health, it looks like an abandoned lot. Weeds everywhere, broken fences. That's the enterobacter and other pathobionts taking over.
What the centenarian?
The centenarian has a super gardener living inside them.
A super gardener that makes its own weed killer and is constantly reinforcing the fence with the help of Akkermansia.
That biological resilience is what seems to keep them going.
So, the million-dollar question then. If I don't have that super gardener naturally, if my genetics didn't give me a gut full of Christensenlacea, Can I hire one? Can we engineer this?
And that is the transition. That's the leap from observation to intervention. And it shifts our focus from who is there to what chemicals are they making.
The metabolites.
We call this the metabolites as medicine approach.
Okay, let's talk about these metabolites, because one of the most exciting ones that came up in the research stack was urolithin A.
Urolithin A is an absolute superstar in longevity research right now. It is getting so much attention, and for good reason.
I've seen the supplements for it starting to pop up online, and the story is that it's connected to pomegranates.
Well, yes and no. And this is the trick that trips a lot of people up. Pomegranates, walnuts, some berries, they contain these compounds called elagitannins.
Elagitan.
But here's the catch. Your body, your human cells, cannot use elagitannins directly. They're just potential energy. They're locked away. You could eat pomegranates all day long, drink gallons of the juice, and it might do absolutely nothing for your longevity.
Because I'm missing the converter, the key to unlock it.
Exactly. You need specific gut bacteria to perform that conversion, to turn the elagitannins into urolithin A. The bacteria are the factory workers. If you don't have those specific bacterial strains on the factory floor, the factory is closed. No medicine gets produced.
That explains so much about why nutritional studies are so messy. You give 100 people pomegranates, half of them get better, half of them don't, and for years, nobody understood why.
Precisely. It's the microbiome variable. We were ignoring the middleman. But for the people who do have the right bacteria, or for people who now take bioidentical urolithin A directly as a supplement, the effects are profound.
What does it do?
It induces a process called mitophagy.
Mitophagy. I love this word. Let's break it down for everyone listening. What is happening inside the cell?
Okay, so imagine your individual cells are like a bustling city. The power plants of this city are the mitochondria. They generate all the energy for everything you do. Thinking, moving, breathing, everything.
The powerhouses of the cell. I remember that from high school biology.
That's the one. But just like real power plants, over time, they get old, they get rusty, they start to leak toxic waste, they become inefficient, and stop producing energy effectively.
And in a normal aging body, do they just sit there clogging up the works and polluting the city?
For the most part, yes. They accumulate. That accumulation of junk, of broken, dysfunctional mitochondria is a core hallmark of cellular aging.
Yeah.
It's like a city filled with abandoned smoking factories.
Okay, so what's mitophagy?
Mitophagy is the city's recycling program.
Ah, so it cleans up the mess.
It does more than that. It identifies those broken down, rusty power plants, it dismantles them piece by piece, and then it recycles the raw materials to build brand new, shiny, efficient ones.
And urolithin A is what triggers this recycling program.
It hits the giant start button on the recycling plant. It sends a signal to the cell that says, hey, it's time to clean this place up.
That sounds incredible in theory, but does it actually work in humans? Is there clinical data?
There is. The clinical trials are very promising. Sources like JAMA Network Open and Nature Aging have published studies showing that urolithin supplementation significantly improves muscle endurance and strength in older adults. It literally counters the age-related decline in cellular energy.
So it's like putting a new engine in an old car.
A whole fleet of new engines in every cell. And it's not just muscles. Other research shows it also reboots the immune system.
How?
It seems to reverse the aging of hematopoietic stem cells. Those are the parent cells in your bone marrow that create all your new blood and immune cells.
So you're getting younger blood and stronger muscles.
That's the promise. All from a metabolite that your gut bacteria can make for you.
If you have the right bacteria.
If you have the right bacteria.
Right. Okay, let's talk about another one of these chemical pathways. Tryptophan. I know this one. It's the stuff in turkey that makes you sleepy after Thanksgiving dinner, right?
That's the popular myth, yes. It's a bit more complicated than that. But in the gut, tryptophan is a building block for something else entirely.
What's that?
Gut bacteria convert tryptophan into a class of compounds called indoles.
Indoles. Now, are these good guys or bad guys?
They are complicated. The classic, it depends.
My favorite kind of scientific answer, it depends.
Well, it's all about balance and context. On the one hand, if tryptophan goes down the wrong metabolic pathway, it can be converted in the liver into toxins like indoxyl sulfate.
And what does that do?
It's very bad. It can damage your kidneys and is associated with chronic kidney disease. So that's the dark side.
Okay, so that's the bad path. What's the good path?
On the other hand, the indoles produced by the right bacteria are absolutely critical for activating something called the aryl hydrocarbon receptor, or RHR.
RHR, what is that?
Think of the AHR as a sensor, or maybe a lock, on the surface of your immune cells, especially your T cells. When indels hit this sensor, when the key goes into the lock, it sends a signal that tells the T cells to calm down.
So it's an anti-inflammatory signal. It's telling the immune system to chill out.
Exactly.
Huh.
It reduces inflammation. It signals for repair of the gut lining. It literally helps fight conditions like arthritis and inflammatory bowel disease.
So the same starting material tryptophan from your diet can either end up frying your kidneys or healing your gut. And the deciding factor is which bacteria are processing it and how.
Exactly. It shows why eating healthy isn't a simple equation. You can't just say eat more tryptophan. It completely depends on the chemical factory you have running inside you.
Which brings us to perhaps the most mind-bending part of this whole stack of research. We've talked about muscles, we've talked about immunity and inflammation. Now we have to talk about the mind.
The gut-brain axis.
Yeah.
This is where we start to question things like personality and even free will.
That sounds pretty dramatic. Free will. But the biology seems to back it up, doesn't it?
It really does. And it starts with the physical anatomy. There's a nerve called the vagus nerve.
Right.
It's not some tiny little thing. It's a physical superhighway, a thick bundle of nerve fibers that connects your gut directly to your brainstem. And it's a two-way street, but a huge amount of the traffic, maybe 80 to 90%, is going from the gut up to the brain.
The gut is talking to the brain more than the brain is talking to the gut.
Overwhelmingly so.
And what kind of information is traveling on that highway? What's it saying?
Neurotransmitters. You know, serotonin, right? The happiness hormone.
Sure, it's the molecule that antidepressants like Prozac target in the brain. They try to keep serotonin levels high to improve mood.
Well, hold on to your seat. About 90 to 95% of your body's total serotonin is produced in the gut, not in the brain.
95%. That seems wildly disproportionate. Why does the gut need that much of the happiness molecule?
Well, on a local level, serotonin does a lot of things, like regulating gut motility, bowel movements. But that massive pool of serotonin and its precursors can absolutely influence the brain.
Wow.
Through that vagus nerve signaling. And specific bacteria directly control its production. There's a particular probiotic strain called E. coli Nissle 1917, we'll talk more about them later, that has been shown to directly influence serotonin availability.
So does this actually change how we feel or how we think? Or is it just a local effect in the gut?
It absolutely changes behavior. There was a study in the journal Nutrients that showed that altering gut bacteria can influence what they called social decision-making.
Social decision-making. What does that even mean? Like who I vote for or who I decide to be friends with?
Maybe not that specific yet, but it impacts things like your sense of fairness, your altruism, your reaction to being treated unfairly.
How do they test that?
They use economic games in these studies. For example, there's one called the ultimatum game.
Okay, how does that work?
I'm given $100, and I have to offer you a split. Let's say I offer you $50.
All right.
That's a fair split you'll probably accept. We both walk away happy.
Right.
But what if I offer you an unfair split? I keep $90 and I only offer you $10.
I'd probably be pretty angry.
Exactly. Now, logically, you should still accept the $10 because it's better than nothing. But many people get so offended by the unfairness that they reject the offer, meaning neither of us gets anything. It's a spiteful move, but it's driven by emotion.
Okay.
That reaction to unfairness, how likely you are to reject the offer out of principle. It seems to be modulated by your microbiome.
Wait,
Yeah.
You are telling me that if I'm playing a game and someone tries to cheat me, my reaction, whether I flip the board in anger or whether I just calmly take the $10, might depend on which bacteria are living in my intestines?
The evidence suggests they play a significant role. It's leading to this idea of the social microbiome. It even raises a crazy question. Are we sharing microbes with the people we're close to in order to share moods and align our thinking?
That is profound. It completely redefines personality. It's not, I'm a grumpy person. It's, my Clostridium is flaring up and making me reject unfair offers.
And here is where it gets dangerous. Stress makes this whole thing a vicious cycle. We know for a fact that psychological stress physically changes the gut.
How? Is it a chemical signal?
It's physical. Stress hormones like cortisol increase intestinal permeability. They literally make your gut leaky. But they also make the blood-brain barrier leaky.
Oh, wow. So the toxins, the LPS we talked about leaking from the gut, they get into the blood, and then the leaky blood-brain barrier lets them cross directly into the brain.
Precisely. Stress opens both gates because you get this horrifying feedback loop.
Let's trace it.
Stress leads to a leaky gut and a leaky brain barrier. That leads to systemic and neural inflammation. That inflammation causes brain fog, anxiety, and depression, which in turn causes more psychological
So
stress.
it's a downward spiral. Stress begets leaky gut, which begets brain inflammation, which begets more stress.
It's a feedback loop from hell. And it suggests that breaking that loop might not be about therapy or meditation alone. It might be about physically fixing the gut barrier.
And that is why scientists aren't just telling us to eat more fiber anymore. They're rolling up their sleeves and getting out the gene editing tools.
Exactly. This brings us to section 4, engineering life. This is the sci-fi stuff becoming tangible reality.
We're moving from just eating yogurt to actively programming bacteria.
We're moving from probiotics, which are basically just wild bacteria we happen to find in yogurt or soil, to live biotherapeutic products, or LBPs.
LBPs, that sounds much more official, more pharmaceutical.
Because it is. These are regulated pharmaceutical-grade living medicines. This is not something you pick up in the health food aisle. These are engineered biological machines.
And to build a machine, you need a chassis, right? Like a car frame you can build on.
Yes. In synthetic biology, you need a base model, a reliable, safe, well-understood organism that you can bolt new equipment onto. And the most famous chassis in this field is a bacterium called Escherichia coli Nissle, 1917.
You mentioned Nissle before with the serotonin connection, but there's a great story behind it, isn't there? Why do we use this one specific bug?
It's an amazing origin story. It goes all the way back to World War I. There was a German army doctor and researcher named Alfred Nissle. He was investigating a horrific dysentery outbreak in a unit of soldiers on the Balkan front.
Okay, so set the scene. World War I trenches. Dysentery is a death sentence for many of these guys.
It's horrific. It's caused by a pathogen called Shigella. People are getting incredibly sick, dying of severe diarrhea and dehydration. And in this one unit, everyone was getting sick, except for one guy, one single soldier.
In the middle of an epidemic, this one soldier is totally fine.
He's completely fine, totally unaffected. So Nissle, being a good scientist, thought, what is special about this man? What's protecting him? So he took a stool sample from this soldier.
Right in the middle of a war zone.
Right there. And he took it back to his lab. And what did he find? He isolated a unique strain of E. coli. It turned out this particular strain was incredibly good at fighting off pathogens. It was aggressive in defense of its host. It was basically a microscopic bodyguard.
So this soldier just happened to have a superbug living in his gut that was protecting him from dysentery.
Exactly. And now, over 100 years later, we use that very same soldier's bacterium as the chassis for modern genetic engineering.
Why that one?
Because it's proven to be safe. It colonizes the human gut really well-meaning. It actually sets up a home there instead of just passing through. And it's genetically easy to hack.
So what are we hacking it to do? Let's get into some of the success stories from the research. What can these engineered bugs do?
Let's start with Parkinson's disease.
Okay, Parkinson's is a brutal neurodegenerative disease. Tremors, stiffness, loss of motor control. It's caused by the death of dopamine-producing neurons in the brain.
Right. So scientists took the Nissle chassis and they engineered it to produce a peptide called GLP-1.
GLP-1, that sounds familiar. Isn't that the basis for weight loss drugs like Ozempic?
The very same. But in addition to its effects on appetite, GLP-1 is also highly neuroprotective. It protects brain cells from dying.
So they taught the bacteria to make this protective drug.
Exactly.
Yeah.
And in the mouse models of Parkinson's, it was incredible. They gave the mice these engineered bacteria. The bacteria set up shop in the gut and became a continuous 247 drug factory.
Pumping out GLP-1.
Pumping out GLP-1, which then entered the bloodstream, crossed the blood-brain barrier, protected those precious dopamine neurons, and significantly improved the mice's motor function.
That is just incredible. So instead of taking a pill or needing an injection every single day, you could theoretically swallow the factory once,
Yeah.
and it manufactures the medicine inside you right where it's needed.
That is the dream. Continuous localized drug delivery. And it's not just for treating diseases. It's also for detection.
You mean like cancer detection?
Yes.
How does that work? Bacteria can't see cancer.
No, but they can taste the environment. Tumors create a very specific chemical environment around them. They're often hypoxic, meaning low in oxygen, and they're acidic because of their weird metabolism.
Okay, so it's a unique neighborhood.
It's a unique neighborhood, and scientists can engineer the bacteria to sense that environment. They put a genetic switch in them that only flips on when they find a place that's both low oxygen and acidic.
And when the switch flips,
It tells the bacteria to produce a reporter molecule.
like a flare gun.
Exactly. A harmless little molecule that is then absorbed into the blood and excreted in the urine. So the bacteria patrol your body, and if they find a tumor, they send a signal out through your urine that you could detect with a simple test strip.
That beats a painful, invasive biopsy any day of the week.
By a mile. And in some more advanced versions, they're not just releasing a reporter. They can be engineered to release a therapeutic payload, a chemotherapy drug, directly at the tumor site, killing the cancer cells while minimizing damage to the rest of the body.
It's a microscopic targeted missile.
That's a great way to think about it.
What about metabolic diseases? The outline mentions PKU.
PKU, or phenylketonuria. It's a genetic condition where people are born without the enzyme needed to break down an amino acid called phenylalanine.
Which is found in almost all protein sources: meat, dairy, nuts.
Everything. So if they eat a normal diet, phenylalanine builds up in their blood and it causes severe brain damage. They have to follow an incredibly strict, difficult, and expensive diet for their entire lives.
So how does engineering help them?
It's beautifully simple, in a way. Scientists engineered bacteria to produce the missing enzyme.
The one that breaks down phenylalanine.
The very one. So you put this bacterial factory in the gut, and it just sits there, digesting the toxic phenylalanine for you before it ever has a chance to be absorbed into the blood. It restores the metabolic function that the patient's own body is missing.
It's like installing a software patch for the human body.
That is a perfect way to put it. But, and this is a huge but, all of this involves gene editing. We're using tools like CRISPR to splice these new genes into the bacteria, and that raises a massive flashing red light for safety.
Right, I was just thinking that. If I swallow these genetically modified superbugs and then I go to the bathroom, do they get into the sewer system? Do they get into the water supply? What happens if they start evolving out in the wild?
That is the nightmare scenario. You're asking the single most important question in this field. We cannot just release engineered organisms into the environment. The risk of what's called horizontal gene transfer is very real.
What is horizontal gene transfer?
Bacteria are very generous with their DNA. They have mechanisms to swap useful genes with each other, like their trading cards.
So our engineered bug could meet a nasty pathogen in the sewer.
And pass on its engineered genes. Imagine if we engineered a bug to be resistant to stomach acid, and it passed that gene to a cholera bacterium. You could accidentally create a superpathogen.
Okay, that's terrifying. So how do we stop it? How do we keep the genie in the bottle?
Biocontainment. We have to build in kill switches.
A kill switch, like in an action movie.
Exactly like that, but biological.
Yeah.
The most common way is to engineer what are called auxotrophies.
Auxotrophies. Break that down for me.
Basically, we genetically cripple the bacteria. We make them dependent on a specific nutrient that they absolutely cannot make for themselves and that they cannot find in nature.
Like what?
A synthetic amino acid, for example. Something that doesn't exist anywhere in the wild.
So the bacteria can only survive if I, the patient, am taking a specific pill every day that contains that unique nutrient.
Exactly. It's a binary on-off system. If they are in your gut and you're taking the pill, they live and they do their job. The moment they leave your body and enter the sewer, or if you simply stop taking the pill, they starve and die within hours. They cannot survive in the environment because their special food isn't there.
That's incredibly smart. It's a biological leash.
It's an absolutely necessary leash. But even with that, manufacturing these things is a massive headache.
Why? I mean, bacteria grow everywhere. They grow on my kitchen sponge without any help from me.
Wild bacteria do. But many of the most beneficial gut microbes we want to use, like Akkermansia, are strict anaerobes.
Meaning?
Meaning oxygen is poison to them. It kills them. So you have to develop, build, and run entire manufacturing facilities that are completely 100% oxygen-free.
You can't just brew them in a vat like you're making beer.
Not at all. And then you have to keep them alive. Aspirin doesn't care if it sits on a shelf for a year in a bottle, but a living drug. Figuring out how to keep it viable through freeze-drying, packaging, shipping, and sitting on a pharmacy shelf is a monumental engineering challenge.
So we have the biology, we have the engineering. Now let's talk about the data, because none of this works if we don't know what to target in the first place for each person.
And this is where we enter the era of the digital twin.
I love this concept. Tell me about the Human Phenotype Project that's mentioned in our sources.
This is a project at the Weizmann Institute in Israel. It is just massive. They are collecting data on a scale that we have never seen before in human history. They're looking at 17 different body systems simultaneously in thousands of people over long periods of time.
What kind of data are they grabbing? It can't just be blood tests.
It's everything. Deep sequencing of microbiomes from the gut, the oral cavity, the skin. They have people on continuous sleep and glucose monitors. They're taking Regular blood tests for thousands of metabolites. They're even taking voice recordings.
Voice recordings, why are they recording people's voices?
Because it turns out that subtle changes in your speech patterns, your pitch, your cadence, your word choice, can be an incredibly early predictor for neurodegenerative diseases like Parkinson's or cognitive decline, years before other symptoms show up.
So they feed all of this. What did the source say? 260 billion data points? They feed that into a computer.
They feed it all into a massive AI model.
Yeah.
And the goal is to create a high-fidelity digital twin of each patient.
So there is a virtual me, a simulation of my biology living inside the Weizmann Institute's servers.
That's the idea. And that virtual you allows for something truly revolutionary, predictive medicine.
How is that different from what we do now?
Right now, what we call healthcare is actually sick care. It's reactive. You get sick, you feel pain, you go to the doctor, and they try to treat the problem that's already there.
Right.
Predictive medicine uses the AI model and your digital twin to look at your trajectory. Based on your microbiome composition and your metabolite levels today, the AI can predict what disease you are on a path to get in 5 or maybe 10 years.
It's like the movie Minority Report, but for predicting heart attacks instead of crimes.
That's a perfect analogy. The AI can see the subtle pattern of biome-aging starting to happen long before you feel any symptoms at all. It might say, your bacteroids levels are dropping and these inflammatory markers are trending up. You are on a clear path to develop cardiovascular disease in 7 years.
That is both terrifying and incredibly useful. So then what do I do with that information?
Well, this is the best part. We then test the intervention on your twin.
On the virtual me.
Yes. Instead of the doctor just giving you vague advice like eat better, the AI can test hundreds of specific interventions on your digital twin. It runs the simulation. What happens if virtual Andrew eats a high-fiber diet? Does that fix the trajectory? No. Okay, what if we add a supplement of Akkermansia? Ah, yes, that works. That puts him back on a healthy path.
So I don't have to be the guinea pig. I don't have to try a diet for 6 months just to see if it works for me. We can test it virtually first.
Correct. It's hyper-personalized nutrition and medicine. No more one-size-fits-all recommendations. It's this size fits you and only you.
This sounds absolutely amazing, but it also sounds incredibly expensive.
And that leads us directly into the ethical minefield. Section 6.
Designing Methuselah.
We have to start by asking the most basic question. Is aging a disease that should be cured?
It's a huge debate. Silicon Valley certainly seems to think so. Others argue that it's a natural and necessary part of life.
If we accept the premise for a moment that we can engineer the microbiome to dramatically slow down aging, maybe even double human lifespans, the next question is, should we?
I mean, my first instinct is to say, of course. Who wouldn't want to live a longer, healthier life?
But you have to think about the societal consequences. Evolution, both biological and cultural, relies on turnover. New generations bring new adaptations, new ideas, new ways of thinking. If everyone lives to be 150 or 200, what happens to our resources? What happens to social progress when the old guard never leaves?
And more importantly, who gets to live to 150?
That is the equity gap. And this technology could create the biggest one in human history. We could see the emergence of a microbiome divide.
I can see it now. The wealthy will be able to afford centenarian microbiome transplants and these custom-engineered AI-designed probiotics.
While the poor are left with what some researchers are calling industrialized microbiomes.
Meaning, what exactly?
Meaning microbiomes that have been degraded by decades of processed foods, pollution, chronic stress, and overuse of antibiotics. Their microbiomes will continue to age faster, making them sicker and leading to shorter lives, while the rich stay biologically young.
It literally creates a biological caste system.
It does. A feedback loop where wealth leads to health, which leads to more wealth. And that's why some researchers are proposing a really radical idea to counter this.
What's that?
Recognizing the human gut microbiome as a common global heritage.
Like the ocean. Or the moon, something that belongs to everyone.
Exactly, or like the major food crops that are essential for humanity's survival. The idea is that the incredible biodiversity of the human gut, especially the rare, diverse microbiomes found in indigenous populations who haven't been exposed to industrial lifestyles yet, should belong to all of humanity, not be patented and owned by a single pharmaceutical company.
So that fair access to these life-extending technologies becomes a matter of human rights.
That's the argument. Because if we lock this knowledge behind an insurmountable paywall, we risk exacerbating inequality on a fundamental biological level.
And then there's the safety regulation we touched on earlier at the FDA.
Yes, the regulators are wrestling with this right now. How do you regulate a drug that is alive? A drug that can change and evolve after you take it. They've created a new category for these live biotherapeutic products, but the rules are still being written as we speak.
Because if you get it wrong,
If you get it wrong and you approve a gene-edited bug that escapes containment and transfers antibiotic resistance to a deadly pathogen, you could start a global plague. The risk of horizontal gene transfer is not trivial.
it's a constant balance, isn't it, between the incredible quest for immortality and the risk of absolute catastrophe.
As is all great science.
So let's try to bring all of this back home. We've gone from the invisible clock ticking away in our guts to the centenarians who seem to defy time, to the chemical factories, the engineered superbugs, the digital twins, and these massive ethical dilemmas.
It is a lot to take in. It changes everything.
It really does. But what's the core takeaway for the person listening right now? Why should you care about this today, years before most of these engineered pills are available?
Because you are a holobiont today, right
Well,
now.
all right.
You don't have to wait for the high-tech engineered bacteria to start taking better care of your garden.
The super gardener from the lab might be in the future, but we can all still weed and water the plants we have right now.
Exactly. Just understanding that fiber isn't just bulk to be passed through, but is essential fuel for the bacteria that keep your gut lining intact. Understanding that psychological stress physically damages that barrier. Understanding that a diverse diet full of different plants leads directly to a diverse, resilient ecosystem.
It shifts the entire perspective from I need to go on a diet to lose weight to I need to feed and support my ecosystem.
That shift in mindset is incredibly powerful. You are the custodian of trillions of lives inside you. Treat them well, and they will treat you well.
And maybe, just maybe, they'll help you live to be 100.
That is the hope.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
Absolutely. The science is moving incredibly fast, and we are just honored to be able to share it with you.
And to our listeners, thank you for being part of this journey with us. If you enjoyed today's episode, be sure to subscribe and follow Stack Studios for more informative content, and support us in Patreon.
Your support is what keeps this exploration going.
Until next time, stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.
Goodbye, everyone. Take care of your gardens.